imagine that right now, inside every single one of your cells, there's this, well, basically a microscopic alien living a completely parallel existence to you.
Yeah, it's a pretty wild thought.
Right. And it has its own entirely separate DNA. It plays by its own biological rules. And it actually has this, personal unbroken history that stretches back literally hundreds of thousands of years.
Hundreds of thousands, yeah.
And the craziest part is you didn't even build it. Like, it's not from your own genetic blueprint. It lives inside you, and it acts as the literal sole power source for your entire existence.
I mean, it honestly sounds like the setup for some sort of sci-fi thriller, right?
Totally, like Invasion of the Body Snatchers or something.
Exactly. But scientifically speaking, it is an absolute reality. You're describing mitochondria.
Right.
And, the reason this whole concept feels so jarring to people is because it fundamentally shatters one of the most, I guess, persistent biological myths we're all taught.
The 50-50 split.
Yes, exactly. The 50-50 split. We all grow up believing that our genetic makeup is this perfectly balanced equation, like exactly 50% from your mom and exactly 50% from your dad.
Right. I mean, that's biology 101.
Sure, but the consensus across all the genetic research and medical case files we're unpacking today is that the 50-50 split is, well, it's a mathematical lie.
A lie.
Yeah. You are actually, at a foundational cellular level, slightly more your mother than your father.
Okay, that is mind-bending. Because if the 50-50 split is just a myth, that completely rewrites how we, calculate inheritance, how we even look at our own family trees.
It changes everything.
So for this deep dive, we're looking at a pretty heavy stack of sources. We've got medical case files detailing this incredibly controversial procedure from 2016, and we're also pulling from this newly released diagnostic framework by MAPASGEN.
Right, the Genetics Research Collective.
Yeah, the ones who focus specifically on mitochondrial health and inheritance. So our mission today, okay, let's unpack this. We want to explore this absolute biological matriarchy that is functioning inside your cells right now.
And it is an absolute matriarchy.
And we also need to understand the catastrophic cascade that happens when these microscopic power plants just fail. Plus, we're going to look at this literal medical heist that bypassed international laws to create the world's first three-parent baby.
The stakes here are they're immense. I mean, we're not just kicking around abstract evolutionary theory today.
No, not at all.
We are talking about the literal physical energy currency that is keeping your organs running while you listen to this. And, arguably more importantly, we're looking at the ethical boundaries that scientists they're actively crossing right now to correct the really brutal errors of human evolution.
Okay, so let's start at the very beginning. Like, what actually happens at the moment of conception?
Yeah.
Because that's where this whole biological matriarchy really establishes its dominance, right?
That's the flashpoint, yeah.
Because we usually picture fertilization as this beautiful, equitable merging of two genetic libraries. But with mitochondria, the mechanics are just ruthlessly exclusionary.
Very ruthless.
So if the sperm is carrying the father's genetic payload, how on earth does it manage to contribute absolutely zero mitochondria to the final embryo? Like, none at all.
So to understand that, you have to look past the basic anatomy of a sperm cell and really dive into this concept called genomic conflict.
Okay.
Think of a sperm cell as a highly specialized delivery vehicle.
Right.
It has this payload of nuclear DNA and then has tail that acts like a biological outboard motor.
To swim to the egg.
Exactly. And to power that motor for this incredibly arduous journey, the mid-piece of the sperm is just tightly packed with the father's mitochondria, and they are burning through energy at an absolutely massive rate.
So the father does have mitochondria in the mix initially.
He does. But when that sperm finally breaches the egg, it doesn't just casually drop its tail and leave the mitochondria at the door. The egg actively destroys them.
Wait, what? It actively destroys the father's mitochondria.
Oh, yeah.
So it's not just like, oh, the tail breaks off and they accidentally get lost.
No. It's a targeted, intentional elimination. Evolutionary biologists call this the resolution of a genomic conflict. See, the father's mitochondria have been running at maximum redline capacity just to win the race to the egg.
Right, they're exhausted.
Exactly, which means they're highly stressed, and because of that, they're prone to rapid mutation. The egg actually recognizes this. So the very moment the sperm enters, the egg's machinery tags the paternal mitochondria with this specific protein called ubiquitin.
Ubiquitin.
Yeah. And in cellular biology, getting a ubiquitin tag is basically receiving a molecular death sentence.
Wow.
It signals the cell's waste disposal units to actively hunt down and completely dismantle every single one of the father's mitochondria.
So the egg literally initiates a microscopic search and destroy mission,
That's exactly what it is.
just to make sure the father's energy cells don't survive. But why? Why is the egg so aggressively territorial about the power supply?
Well, because if maternal and paternal mitochondria were allowed to coexist and, replicate inside the same embryo, it would basically trigger a cellular civil war.
Oh, wow.
Yeah, mitochondria have their own DNA, which we mentioned. That means they replicate on their own schedule. So if you have two different populations of mitochondria competing for the exact same resources inside one cell,
They just fight each other.
right, they stop focusing on producing energy for the developing fetus, and instead they just focus on out-replicating the other side.
That sounds disastrous.
It is. The resulting inefficiency would be fatal to the embryo. So evolution just solved this by enforcing a strict monopoly. The mother's mitochondria completely take over, and the father's are annihilated.
Annihilated. Which means that every single mitochondrion in your body right now, like the ones powering your brain, your heart, your muscles, as you listen to this, they came exclusively from your mother's egg.
100% of them.
And hers came from her mother. And so on and so on. It just creates this completely unbroken, purely female lineage. I could totally get why the MAPASGEN researchers coined the term molecular matriarchy.
It really is, and this strict maternal inheritance gives geneticists this incredibly powerful tool because think about nuclear DNA, the DNA in the nucleus of your cells. That's a messy combination of both parents.
Right, it's a mix.
Yeah, it gets shuffled and recombined with every single generation. It's literally like shuffling a deck of cards. But mitochondrial DNA, it doesn't shuffle.
Just copied.
Exactly. It's cloned and passed down directly. It only changes through these very rare random mutations that happen at a highly predictable rate. And because that mutation rate is so predictable, mitochondrial DNA essentially acts as a molecular clock.
Oh, so they can trace it back.
Right. By comparing the mitochondrial DNA of people alive today, scientists can wind that clock backward.
And when they wind that clock all the way back, the genetic consensus points to just one single woman, right? Mitochondrial Eve.
Correct. The mathematics dictate that this unbroken chain of maternal inheritance converges on a single human female. She lived in Africa approximately 200,000 years ago.
Just one woman. That's wild.
Well, to be clear, she wasn't the only woman alive at the time. But her specific mitochondrial lineage is the only one that survived to be present in every single human being alive today.
That realization is just staggering to me. Like you sitting there listening to the exact same cellular machinery that powered mitochondrial Eve 200,000 years ago, the stuff that allowed her to metabolize food and survive the Bronze Age and everything else, it is currently spinning the microscopic turbines in your cells right now.
It's a profound
It
connection.
really is. But okay, here's where it gets really interesting. Because if we know this system of inheritance is so stable, this absolute clone passed down through millennia, what happens when there's a manufacturing defect?
That's the dark side of it.
Right. If a flaw gets introduced into that maternal line, the mother has literally no choice but to pass that flaw down to every single one of her children.
Yes. And that is the devastating vulnerability of the molecular matriarchy. When the cellular battery fails, the results are catastrophic. And the MAPASGEN diagnostic framework uses this highly effective analogy to explain the difference between your nuclear DNA and your mitochondrial DNA.
The electric car one.
Well, let's use the city one first because it scales so well.
Yeah.
Think of your body as a sprawling, modern city.
Okay. So the nuclear DNA, the genetic material we get from both parents, that's like the architectural blueprint for the city.
Exactly.
It determines where the skyscrapers go, the layout of the streets, the bridges. It dictates your height, your bone structure, your eye color.
Precisely. You could have a flawlessly designed city. The architecture is brilliant. The infrastructure is perfectly laid out. But the mitochondrial DNA represents a completely different system. It's the city's power grid.
Oh, yeah.
And the power grid doesn't care about the architecture. It doesn't decide what the buildings look like or who gets to live in them. Its only job is to keep the lights on. So if the power lines fail, it doesn't matter how beautiful a city is. The hospitals shut down, the traffic lights go dark, the water stops pumping, the entire city collapses.
And mapping that back to the human body, the organs that require the most electricity are the ones that are going to collapse first.
Absolutely. The brain, the heart, the nervous system.
So how does that energy actually work at the micro level?
So When we talk about cellular energy, we are talking about a very specific molecule called ATP, adenosine triphosphate.
ATP, right?
Think of APP as a tightly coiled microscopic spring. Your mitochondria act as the power plants that coil those springs. So whenever a muscle contracts or a neuron fires in your brain, it does so by snapping one of those springs to release energy.
Okay, I like that visual.
And your brain alone, despite being only about 2% of your body weight, it consumes roughly 20% of your body's total ATP. It is a massive energy sink.
So if a child inherits defective mitochondria, their cellular power plants physically cannot coil enough of those ATP springs to meet the demand.
They just can't keep up.
Which brings us to the specific medical case files we're looking at today. The devastating reality of Leigh syndrome.
Yeah. Leigh syndrome is a severe neurological disorder, and it perfectly illustrates the cruelty of mitochondrial mutation. Because of how the disease manifests, a child with Leigh syndrome is often born looking perfectly healthy.
Because the blueprint was fine.
Exactly. The architectural blueprint of the city was flawless, but within a few months, as the child's developing brain and central nervous system began to demand exponentially more energy, the defective power grid just fails.
The neurons literally start starving to death.
They starve and they begin to die. The medical reality is this rapid, progressive loss of mental and motor abilities. The brain develops actual lesions because the tissue is suffocating from a lack of cellular energy.
That is horrifying.
It is. The child loses the ability to suck, to hold their head up, to breathe independently. And because this is a flaw in the foundational power source of the cell, there is no cure.
And historically, because of these strict rules of maternal inheritance we just talked about, this was basically a biological trap.
A total trap.
Right. A woman carrying the mutation for Leigh syndrome in her mitochondria faces an impossible reality. Her nuclear DNA might be perfectly healthy. The father's DNA is perfectly healthy. But because the sperm's mitochondria are destroyed at conception, she alone provides the cellular power grid to her offspring. Every single child she has will inherit that flawed grid.
It creates an agonizing scenario for families. I mean, historically, women carrying these severe mitochondrial mutations were given only a few options, and all of them were grim.
Yeah.
They could rely on egg donors and abandon a genetic connection to their child entirely, or they could roll the biological dice, knowing there was an overwhelming probability their child would suffer this progressive, fatal energy failure.
But if we know exactly where the flaw is, like if we know the architectural blueprint is perfect and only the power grid is failing, the logical workaround would be finding a way to keep the mother's blueprint, but just scrap her defective power grid.
In theory, yes.
But was anyone actually capable of attempting something that radical on a microscopic scale?
They were, but not without crossing some massive regulatory and ethical boundaries, which brings us to the really controversial actions of Dr. Zhang.
Right. The fertility specialist in New York.
Exactly. So by 2016, Dr. Zhang was treating a couple who carried the Leigh syndrome mutation. And the mother had already experienced the tragedy of this inheritance firsthand. They had lost 2 children to the disease.
Oh, that's just heartbreaking.
It is. The lottery of natural inheritance was just no longer an option for them.
And Zhang knows that the theoretical technology to perform a mitochondrial swap exists. But he also knows that the FDA in the United States has strictly banned any procedure that involves modifying the germline.
Right, the genetic material that can be passed down.
Yeah, because the regulatory body has considered the procedure far too experimental, with completely unknown long-term consequences.
So, faced with this regulatory wall in the U.S., Zhang made an unprecedented decision. He didn't abandon the procedure. He literally packed up his medical team, all the specialized equipment, his patients, and he moved the entire operation across the border to a clinic in Mexico.
He just left.
He explicitly stated he went to a jurisdiction where there were, quote, no rules regarding this specific type of genetic modification.
It is essentially a high-stakes, cross-border medical heist. Like, he circumvented the federal oversight of his own country to attempt a procedure that had never resulted in a living human being before.
Never.
So how exactly did they execute this? Because I'm trying to wrap my head around it. How do you swap out the power grid of a single cell before a human is even formed?
It's incredibly delicate. The procedure is formally known as mitochondrial replacement therapy, but the specific technique Zhang used is called spindle nuclear transfer.
Spindle nuclear transfer, okay.
And it requires genetic material from 3 distinct individuals: the father, the mother carrying the mutation, and a healthy female donor.
Okay, let's walk through the exact mechanics of this, because the precision required is just it's hard to fathom.
Right. So first, they harvest an egg from the healthy donor. Now, this egg has a perfectly functioning mitochondrial power grid, but it also contains the donor's nuclear DNA.
Her blueprint.
Right. So using a microscopic glass pipette, which is essentially a needle thinner than a human hair, they pierce the donor egg, extract its nucleus, and they discard it.
So they've essentially, using the car analogy now, they've taken the driver out of the car but left the pristine engine running. They have an empty biological shell powered by healthy mitochondria.
That's a great way to put it. Simultaneously, they harvest an egg from the mother. They use another micropipette to carefully extract her nucleus, her core genetic identity, the blueprint. And they inject the mother's nucleus into that hollowed-out donor egg.
But wait, when they suck the mother's nucleus out of her egg, doesn't the pipette inevitably pull up some of the fluid surrounding it? And isn't that fluid swarming with her defective mutated mitochondria? How do they prevent carrying the disease right over into the new, clean egg?
You've hit on the most precarious part of the entire operation. It's a phenomenon known as mitochondrial carryover. Because it is physically impossible to extract the nucleus with 100% purity. A small number of the mother's mutated mitochondria will inevitably hitch a ride into the donor egg.
So they're bringing some of the bad batteries with them.
Exactly. The scientific gamble here is that if they can keep that carryover to under 1 or 2%, the overwhelming majority of the healthy donor mitochondria will outcompete the defective ones, and the disease will never manifest.
Wow, so they transplant the mother's nucleus into the donor's power grid, creating a newly synthesized egg. Then they introduce the father's sperm to fertilize it.
Right.
And the egg, following those ancient rules of genomic conflict we talked about, goes ahead and destroys the father's mitochondria.
Like clockwork. And the resulting embryo begins to divide. So in September 2016, the result of that microscopic surgery and that regulatory evasion was born. A boy named Abbas al-Hassani.
Abbas.
He became the first human being on record to be born carrying the DNA of 3 different people.
3 different people.
He possesses his father's nuclear DNA, his mother's nuclear DNA, and the donor's mitochondrial DNA.
And looking at the medical follow-ups in our sources, the procedure was an absolute biological triumph. Like, Abbas al-Hassani is alive, he is healthy, and the Leigh Syndrome was successfully bypassed. He survived purely because his parents and his doctors fundamentally altered the recipe of his cellular makeup.
It was a massive medical breakthrough, but of course it immediately triggered an ethical firestorm.
Oh, I'm sure.
Because while the medical community celebrated the birth of a healthy boy, ethicists and regulatory bodies around the globe recognized that a really profound line had just been crossed. Zhang hadn't just cured a disease, he had engineered a human chimera.
Right. And let's examine that pushback presented in the sources, because it's not just, like, reactionary fear. If you play devil's advocate for a second, by splicing the nucleus of one woman into the cellular body of another, we have taken a literal scalpel to the sacred, untouched lineage of Mitochondrial Eve.
We severed a chain that ran perfectly intact for 200,000 years.
Exactly. And the primary concern isn't really about the immediate success, is it? It's about the unknown variables 50 years from now.
Modifying the human germline has always been considered the ultimate biological taboo, because the fear is that we simply do not know enough about the intricate crosstalk between the nucleus and the mitochondria.
They have to communicate.
They do constantly. So what happens when the mother's nuclear DNA tries to send a signaling protein to a mitochondrial network that evolved in a completely different genetic environment?
Will it even understand the signal?
Right. Will that communication break down when Abbas is 30 years old?
Yeah.
Will it trigger some sort of early onset neurodegeneration? The critics argue that we are running an uncontrolled experiment on a human life.
And beyond the physiological unknowns, there's a huge societal argument here. If we are perfectly comfortable circumventing federal regulations to swap out a cellular power grid today, how do we hold the line tomorrow?
The slippery slope.
It feels like the ultimate slippery slope. If we can engineer an egg to remove a mitochondrial defect, what stops a clinic from engineering an egg to ensure a specific eye color? Or, tweaking the genome to optimize for height or IQ? We're opening the door to the complete commodification of human biology, like an auto dealer's website configurator for babies.
It's a very common, very visceral societal objection. However, the MAPASGEN researchers and the geneticists who defend the procedure, they draw a very stark, scientifically grounded line against that specific fear.
Okay, what's their defense?
To understand it, we have to go back to our city power grid analogy and look at the actual numbers, the scale of what we're talking about.
The difference between the nucleus and the mitochondria.
Exactly. Your nuclear DNA, the architectural blueprint that determines your traits, your appearance, the complex variables of your personality, that genome is massive. It contains roughly 20,000 genes.
20,000, okay.
Now, the mitochondrial DNA, the entire power grid, it is coded by exactly 37 genes.
Wait, 37? Just 37 genes out of 20,000?
That's it. It is essentially an invisible code, and the crucial distinction here is what those 37 genes actually do.
They just make power.
Exactly. They do not dictate intelligence. They have absolutely zero influence on whether a child will be introverted or extroverted, athletic or artistic. Those 37 genes are highly specialized, purely utilitarian instructions for one single task, synthesizing the proteins necessary for the electron transport chain to produce ATP.
So they don't design the city at all. They literally just manufacture the copper wire that keeps the electricity flowing.
Precisely. So the MAPASGEN defense is that swapping out those 37 genes isn't altering the child's fundamental identity. And it's not moving us toward designer babies. It is purely a mechanical fix to ensure the child survives long enough to actually express their identity.
Wow. I mean, that is the core ethical philosophy presented in these sources. The proponents argue that allowing a family to repeatedly birth children doomed to an agonizing, slow death by suffocation at the cellular level, simply because we're afraid of a theoretical slippery slope regarding 37 utilitarian genes, is the true ethical failure.
That's their stance. When a mechanical fix exists to prevent profound human suffering, they argue we have a moral imperative to use it.
Yet, despite that really compelling defense, the global legal landscape remains deeply fractured, doesn't it?
Oh, very much so.
Yeah.
According to our analysis, while places like the UK, Australia, and Ukraine have actually legalized and established rigorous frameworks for this therapy, the rest of the world is paralyzed. The US FDA still flat out refuses to greenlight the procedure.
Which just creates a system of medical tourism for the desperate. Families who have the money will continue to seek out jurisdictions with looser laws, exactly like Dr. Zhang did.
Right, and that lack of global consensus doesn't stop the science. It just pushes it into environments with less oversight, which you could argue increases the risk to the actual patients.
Yeah. And amidst all these grand regulatory debates, there is the deeply personal reality of the child. Like, Abbas al-Hassani is going to grow up. How does a child conceptualize his own origin when he realizes he literally has two biological mothers?
It's a heavy burden.
How do you explain to a child that their very existence required severing an ancient evolutionary chain?
Well, the sources actually offer a really profound perspective on how to frame that narrative. The psychological guidance suggests moving away from the cold terminology of clinical genetic engineering and focusing entirely on intent.
Okay, how so?
A child born of this procedure can be told, your parents understood that nature was going to fail you. They loved you so intensely before you even existed that they marshaled the absolute pinnacle of human scientific achievement to ensure you could live.
Oh, wow.
Yeah. It reframes this sterile laboratory procedure as an act of profound parental devotion.
That is a really powerful way to reconcile the science with the humanity of the situation. But I want to pivot here because we spent a lot of time discussing these really rare catastrophic mutations and high-stakes medical heists.
Right, the extreme cases.
Yeah. I think we need to ground this back in the reality of you, the listener. How does this molecular matriarchy function in a normal, everyday context? What happens when you don't have a massive mutation like Leigh Syndrome, but your internal power grid is still kind of faltering?
And that brings us directly to the MAPASGEN energy audit. Because mitochondrial health is not this binary state of either perfect function or catastrophic failure. It exists on a vast spectrum.
Like a sliding scale.
Exactly. You can have a power grid that is technically intact, but it's operating at a severely diminished capacity due to environmental factors or just subtle genetic variations.
So, practically speaking, does every listener who is considering starting a family need to rush out and demand a full sequencing of their mitochondrial DNA?
No, the MAPASGEN framework suggests that universal sequencing isn't necessary. But targeted awareness is absolutely critical. They provide a specific checklist of red flags to look for within your family history. And remember, because of the molecular matriarchy, you are looking exclusively at the maternal line.
So you examine your mother, your maternal grandmother, your maternal aunts.
Exactly.
What specific patterns are we looking for in that maternal line then?
Well, the most obvious flag is a history of unexplained early infant mortality that seems isolated to the female lineage.
Right.
But there are more subtle indicators, too. For example, there's a mitochondrial disease called Leber hereditary optic neuropathy. It presents as sudden, unexplained vision loss, typically in young adulthood.
Just sudden vision loss.
Yeah, tracking through the mother's side. Another major indicator is a pattern of chronic progressive muscle weakness or severe exercise intolerance that, again, tracks strictly through the maternal line.
Okay.
If those specific flags exist, the standard genetic panels offered by fertility clinics, which primarily just look at nuclear DNA, they are insufficient. You must specifically request a mitochondrial screening.
Good to know. But let's assume the listener reviews their maternal history and the genetic line is perfectly clean. They inherited a flawless, unmutated power grid from Mitochondrial Eve. The MAPASGEN audit makes it very clear that having a perfect genetic baseline does not guarantee optimal energy.
Right, not at all.
Yeah.
Your daily function is heavily dictated by how you treat your cellular machinery. They refer to this as environmental exploitation.
Environmental exploitation.
Yeah. Because your mitochondria are highly sensitive organelles. Even without a single genetic defect, you can force your power grid into a state of chronic suppression through poor lifestyle inputs.
And the checklist details how our modern environment is uniquely hostile to mitochondrial function. We are talking about chronic disruption of the circadian rhythm.
That's a huge one.
Yeah. Your mitochondria rely on light signals to know when to ramp up ATP production and when to initiate cellular repair. If you are blasting your retinas with artificial blue light at midnight and sleeping erratically, you are literally scrambling the signaling pathways of your power plants.
You're confusing them. And the result of that scrambling is often profound, unexplainable exhaustion. The diagnostic framework differentiates this from normal tiredness.
How so?
Well, if you're experiencing chronic fatigue that is entirely unrelated to physical exertion, meaning you wake up exhausted and remain exhausted even if you haven't moved from your desk all day, that is a massive clinical indicator that your mitochondria are struggling to convert fuel into ATP.
Now, when people hear that they have a cellular energy deficit, I feel like the immediate cultural instinct is to look for a quick, ingestible fix.
An energy drink, a pill.
Exactly. The supplement industry has built an absolute empire on promising to boost your cellular energy. You walk into any health store, and the shelves are lined with proprietary blends claiming to supercharge your mitochondria. So, looking strictly at the MAPASGEN audit, what is grounded in actual biochemical science and what is just expensive marketing fluff?
The audit is ruthless in cutting through that supplement noise. Many over-the-counter energy boosters rely on heavy stimulants like high-dose caffeine or synthetic compounds that simply flood your central nervous system with adrenaline.
They just trick your brain.
Right. It gives you a temporary jolt, but it does absolutely nothing to repair or support the actual mitochondrial machinery. In fact, overstimulating a damaged power grid can actually accelerate its degradation.
You're just revving a broken engine. So what does the science actually support for genuine mechanical mitochondrial maintenance?
The MAPASGEN research highlights two highly specific nutraceuticals that have a direct, scientifically validated role in the production of ATP, coenzyme Q10 and L-carnitine.
Okay, let's break down the actual mechanisms of how these work, because I think understanding the how is crucial to realizing why the other supplements are useless. Let's start with coenzyme Q10.
To understand CoQ10, you have to visualize how mitochondria actually generate power. Inside the mitochondrion, there is a process called the electron transport chain.
Okay.
Think of it like a microscopic bucket brigade. You have a series of protein stations embedded in the mitochondrial membrane. As electrons are passed down this chain from one station to the next, the energy released is used to pump protons across the membrane.
Create a charge.
Exactly, creating a charge differential that ultimately coils that ATP spring we talked about.
And where does coenzyme Q10 fit into that bucket brigade?
CoQ10 is literally the specialized worker carrying the bucket between the stations. It is a lipid-soluble molecule that ferries electrons from complex amine complex 2 down to complex 3 the chain.
So it's moving the energy down the line.
Yes. And if your body is deficient in CoQ10, which naturally happens as we age, or as a side effect of certain medications like statins, the bucket brigade physically slows down. the electrons back up, the protons don't get pumped, and your overall production of ATP just plummets.
Makes sense.
Supplementing CoQ10 directly restocks the workforce required to keep the electron transport chain moving efficiently.
That makes perfect mechanical sense. What about the second one, L-carnitine?
So if CoQ10 is the worker on the assembly line, L-carnitine is the border patrol controlling the raw materials.
It's the border patrol.
Yeah. Your mitochondria generate energy by burning fuel, primarily glucose and fatty acids. But fatty acids cannot just wander into the inner matrix of the mitochondrion on their own. They require a highly specific transport mechanism to cross that inner membrane.
Ah, so L-carnitine acts as the shuttle.
Precisely. L-carnitine binds to long-chain fatty acids and literally shuttles them across the mitochondrial membrane into the matrix where they can be oxidized and burned for fuel.
So without it, they can't get the fuel.
Exactly. If you lack L-carnitine, your mitochondria are essentially surrounded by fuel that they cannot access. They starve while swimming in resources.
Wow.
So if a patient is presenting with that non-exertional fatigue, strategically supplementing CoQ10 and L-carnitine provides the exact mechanical support the power grid needs. It bypasses the superficial stimulation of caffeine and actually fixes the machinery.
It is incredibly empowering to realize that while we are bound by this ancient molecular matriarchy, we aren't entirely passive passengers. Like, we have the biochemical understanding to actively manage and repair the system.
That is the ultimate takeaway from the MAPASGEN project, and really it's the synthesis of everything we've explored today. Their guiding philosophy is explicitly stated: do not fear technology, fear ignorance.
I love that.
Whether we are talking about targeted nutritional support to fix a sluggish electron transport chain, or utilizing unprecedented microscopic surgery to bypass a fatal genetic mutation, the goal of biological science is to provide illumination in the genetic labyrinth. It exists to offer us agency where nature historically only offered tragedy.
We have covered a massive amount of ground today. I mean, we started by exploring the brutal resolution of genomic conflict at the moment of conception, realizing that the destruction of paternal mitochondria connects every single one of us to a singular female ancestor who lived 200,000 years ago.
Mitochondrial Eve.
We examined the catastrophic failure of the cellular power grid in Leigh Syndrome, and we traced the path of a high-stakes medical heist to Mexico that resulted in the world's first three-parent baby. We've looked at the profound ethical debates surrounding the modification of the human germline, and we brought it all back to the daily biochemical maintenance of your own internal power plants.
It is a perfect encapsulation of how rapidly our understanding of human biology is accelerating and the really profound responsibilities that come with that knowledge.
It is. And as we close out this deep dive, I want to leave you, the listener, with one final lingering thought to explore on your own. It builds directly on the mechanics of inheritance we established at the very beginning. We spent a lot of time discussing Abbas al-Hassani, the child born in 2016, from 3 different DNA sources.
Right.
Abbas is a boy. And because of the unyielding laws of the molecular matriarchy, because the sperm always drops its tail and the egg destroys the paternal mitochondria, Abbas cannot pass his unique genetic makeup onto his children. His designer DNA, his transplanted power grid, is a biological dead end. It stops with him.
It does.
But what happens when the first female three-parent baby is born? Because it is inevitable. When a girl is born utilizing donor mitochondria to save her from a genetic curse, she will carry that artificial transplanted power grid in her own eggs. When she grows up and decides to have children of her own, the rules of the matriarchy dictate that she will pass that donor mitochondria on to the next generation.
And they will pass it to the next.
Exactly. For the first time in 200,000 years, humanity will not just be curing a single disease in a single patient. We will have permanently grafted a brand new synthetically engineered branch onto the ancient, unbroken evolutionary tree of Mitochondrial Eve. What does it mean for the future of our species when we step completely out of the shadows of natural selection and we become the active, deliberate authors of our own lineage?